I don't know if you can share, if you can see now my screen. I don't know if you can share, if you can see now my screen. I don't know if you can share, if you can see now my screen. I don't know if you can share, if you can see now my screen. I don't know if you can share. Okay, can you see the screen now? Okay, can you see the screen now?
No, doctor. Still loading.
Still loading. Okay. Okay, can you see the screen now?
No, doctor.
I don't know what is happening with
Still
Zoom.
loading.
Still loading. Okay. Okay, can you see the screen now?
No, doctor.
I don't know what is happening with
Still
Zoom.
loading.
Screen sharing. Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No, doctor.
I don't know what is happening with
Still
Zoom.
loading.
Screen sharing. Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No, doctor.
I don't know what is happening with
Still
Zoom.
loading.
Screen sharing. Oh, still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No,
I
doctor.
don't know what is happening with
Still
Zoom.
loading.
Yes,
Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No,
I
doctor.
don't know what is happening with Zoom.
yes,
Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No,
I
doctor.
don't know what is happening with Zoom.
yes,
Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No,
I
doctor.
don't know what is happening with Zoom.
yes,
Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No,
I
doctor.
don't know what is happening with Zoom.
yes,
Still loading. Okay. Okay, according to the Zoom, I'm sharing the screen. But
No,
I
doctor.
don't know what is happening with Zoom.
yes,
Still loading. Okay. Okay,
Okay. Okay. Oh. Okay. Oh. Okay. Oh. Okay. Oh. Okay. Oh. Okay. Oh. Sorry about these technical problems. Okay. Oh. Sorry about these technical problems.
Still no. Still no.
Hello, testing 1, 2, 3. Can everyone hear me? First of all, we would like to apologize for the minor technical issue, and we appreciate the understanding from our dear panelists, Dr. Tino, and all of our participants. Now, we will continue our webinar today. Dr. Tino, the floor is yours.
Okay. I have the micro activated now. So I will try to present now, okay? Okay.
Okay, sir.
Okay.
Loud and clear.
Okay. Sorry, sorry for the problems. Okay,
It's
so now
okay.
I'm trying to share this. Okay. So I guess that now you can see my screen now, right? Okay.
Yes.
Okay. So I start my presentation. Well, I'm sorry about the disturbance and the technical problems. Well, so I also wanted to start. Thank you very much for the UITM Saba to give me the opportunity to share with you the advances of the project I'm currently developing in collaboration with several researchers from UITM Saba, and a special thanks to the students Castino and Winnie that have been like managing this presentation. I'm sorry for the technical problems, but I hope that from this moment we can go smoothly. So the title of my presentation is the Project Casobiota, some background and recent advances on the project. I will start presenting a bit about me. As your colleague said during the presentation, I have my degree or my Bachelor in Science in Biology in the University of Santiago de Compostela in Spain, and I did my PhD in Biology in the same university. And currently I'm working as a Maria Sklodowska-Curie Fellow in the University of Vigo in Spain. One of the things that define me in regarding research is as I love metal-rich soils and the ecosystems and the plants especially. Either they were on temperate areas, as you can see in the top photography, or in tropical regions such as Sabah, as you can see in the photograph at the bottom. During my career, I have been working on metal-rich soils and species in different places. For example, my PhD thesis, it was ideal during my PhD thesis with the microevolution of this Mediterranean shrub, is the photograph on the left, called Cistus ladanifer. It's a shrub from the Mediterranean, Spain, Portugal, and Northern Africa. And it grows on metal-rich soils, and I studied the microevolution of this plant on metal-rich soils, the origin of the populations growing on metal-rich soils, and the adaptation to those soils in this plant. Then, as a postdoc, I worked in one research center of the SESI in Spain, and I worked on nickel hyperaccumulators, some species, plant species that are able to accumulate in their leaves huge amounts of metals. And during my first postdoc, I developed, I increased my knowledge about hyperaccumulators and soil properties. And during my second postdoc, that was developed in the University of Lorraine, the subject of my second postdoc was the restoration of nickel-rich technosoils from tropical areas. And during that research, I learned about ecological and soil biological parameters. And currently, I'm working on the Casobiota project. This project is funded by the European Union via the Maria Sklodowska-Curie program. The complete title of the project is Exploration of the Soil Biota Associated to Casuarinaceae trees from tropical ultramafic areas. Apart from funding from the European Union, I am developing this project or parts of this project in collaboration with researchers from the UITM Saba. And for that, we received an RPG, our research partnership grant by the UITM that funds some researchers from School of Applied Science, Faculty of Applied Science in UITM Saba, and a team directed or led by Dr. Lo Chor-White. So let me give you some definitions about the title of the project. As you can see, the project is Exploration of Soil Biota Associated to Casuarinaceae trees from tropical ultramafic areas. So let's develop my research or my presentation. First, I will give you some definitions of some difficult terms I presented on the title of the project. Then I will give you some outlooks of previous research that bring me to this project, and I will finish my presentation with some recent results that we have obtained during last months. I will start talking about ultramafic soils. What are ultramafic soils? These soils are special soils that are occupy a small percentage of Earth's surface, around 1% of Earth's surface, and they are the result of Earth tectonics. In this map, in this figure, you can see like the current distribution of the continents. But you probably know, I'm sure you know that the Earth's surface has changed along the geological history of our planet. And you know that due to tectonics, the continental plates are moving, are floating on the mantle, and they are moving on surface. So in some continents are getting closer, some continents are separating, and sometimes the continents, they crash. And in those places where two continents crash, in the place where these continents contacted, we can find ultramafic soils. These soils are developed from ultramafic rocks. These ultramafic rocks are derived from Earth's mantle. I mean, ultramafic areas or ultramafic soils are very special because the rocks that form these soils, they are not from the Earth's surface. They come from inside, from the Earth's mantle, but due to some movements on the planet during the geological history of Earth, these materials that are from the Earth mantle, from around like 20 or 30 kilometers inside the Earth, now are on surface. And the composition of these rocks is quite special. They are, the ultramafic rocks, they are very rich on mafic minerals. Mafic means dark minerals such as olivine or pyroxene, and they have high concentrations of iron, especially magnesium, and other metals. There are two main ultramafic rocks. We have the peridotites, is the image on the left, and depending of metamorphism, that is a process that some rocks get transformed into others. The peridotites can transform into serpentinites, that are the rocks on the photo on the right. As you can see, either the peridotite and the serpentinite, they have very dark color, they are massive, and they have, apart from the color, they have special properties. The ultramafic areas with these rocks are quite abundant. I told you before that ultramafic areas occupy less than 1% of Earth's surface, but they are quite common, they are quite abundant, as you can see in the map, in Southeast Asia. We have some areas in India, in Burma, in Thailand, Vietnam, but Philippines, Sabah, some parts in the south of Kalimantan, and then on other islands of Indonesia, they have huge surface of ultramafic areas. In this other map with more detail, you can see in the red areas are the areas covered with ultramafic soils in Sabah. We have like a line from the northwest to the southeast that begins. We have Malawali Island in the north of Sabah, and then we have some places around Kinabalu Mountain, and the main ultramafic areas are in Meliau Ranch, Bidu Hills, Montawai, and continue until Mount Silam near Lahad Datu. As I told you that ultramafic rocks, they have special properties, and they have a composition that is quite different from the other soils. So the soils that derive from ultramafic rocks, they are very toxic to plants because they don't have a lot of nutrients. They lack phosphorus, they have potassium, they have other essential nutrients. Moreover, they have an excess of magnesium. Magnesium is an element that is important for plant metabolism, but when we have much more magnesium than calcium, the magnesium gets toxic for the plants. And moreover, these ultramafic soils, they are rich in metals, especially nickel, but also chromium and cobalt. So together, these conditions make these soils very toxic or difficult for the plants to grow. This is why I put this death symbol here. But in contrast, in spite of this toxicity, the ultramafic soils are also living evolutionary labs. You can imagine that the ultramafic soils, they are very different from the other soils around there. So when a plant colonizes ultramafic soil, it is subjected to different selection pressures. So they have to adapt to the soils, and they develop, during the evolution to grow on these soils, they develop adaptation strategies, and they gave birth to new species. So ultramafic soils, apart from toxic soils, they are especially rich on endemic plants. So we can say that ultramafic soils are biodiversity hotspots. One example of that is the presence of hyperaccumulators, plants that are able to take this nickel from the soil, especially nickel, and accumulate on the leaves in very high concentrations. The image you have on the left are several photos from the hyperaccumulator species Phyllanthus valgoi that it grows on Sabah. And you can see a detail of it can be it's a big shrub or small tree. It can reach like several meters height. It had small leaves, and then when you cut the skin of the tree, the bark, you can see that it releases green phloem, that it has a lot of nickel, about 20% of nickel. These photographs are from a paper from Mandarin and collaborators. And as I told you, ultramafic areas, they are biodiversity hotspots, but because they are rich in metals. So humans are also interested on ultramafic areas, and we have been extracting. These are some photos from different places in Indonesia. The humans, the people, has been extracted metals from ultramafic soils, especially nickel, but also chromium. And mining ultramafic rocks makes the plant colonization even more difficult. No? As I told you, ultramafic soils, they don't have a lot of nutrients, they are toxic, but when we do the mine, we remove the first layer of soil where most nutrients are, we compact the soil, we degrade the plant communities, so the recolonization or the revegetation of ultramafic soils is very difficult. What we need to recover these soils? Well, we need to restore these areas. We can work like in two main strategies. First of one, we need to create, as we have lost most of the nutrients of the soil because of mining, we need to bring some nutrients to the soil that allows when you reach a minimal amount of nutrients in the soil, you make possible that plants colonize and start to recover the community. So for this restoration, you can follow two approaches. One of them is the use of amendments. I mean, to bring industrial byproducts or compost or materials that are rich on nutrients, and you add them to the soils to bring this first set of nutrients that allow the plants to colonize. Or you can choose, or you can select pioneer or nurse species. Pioneer species are plants that are able to colonize the mines even if you don't plant them. They are able to colonize by themselves. And sometimes these plants can be nurse species. A nurse species means that the plant, when it grows, it creates conditions that allow other plants to colonize. This image I present in this slide. This is a book from New Caledonia. New Caledonia is east of Australia. It's an island that it's part of France, where they have been developing nickel mining since the beginning of the 20th century, and provoking an important environmental impact. So they have been doing an intense work identifying species that can be used for recovery and for rehabilitation. But New Caledonia is an exception. No, normally we don't know a lot. We don't know a lot. We don't know not a lot about which species we can use for revegetation. So this photograph is my previous postdoc, my first time in Sabah. It was in 2016, and I came to Sabah to work in a project dealing with the use of plants from degraded ultramafic areas to know which plants grow on degraded ultramafic areas and how we can use them to restore ultramafic soils. During that postdoc, I came several times to Sabah, and in one of my visits, I stayed in a place close to Mammoth Copper Mine. In this photo is a satellite photograph of Mammoth Copper Mine. Mammoth Copper Mine, if you don't know where it is close to Ranau village. It's north of Ranau. Ranau will be like in the south, below this image. So Mammoth Copper Mine is a huge mine that was exploited from the 70s to the 90s, and the mine is closed now. And it has a big lake full of acid water and a lot of soils that are, as you can see, the soils don't have a lot of vegetation on them because of toxicity. And when I visited Mammoth Copper Mine, I was not interested on the copper mine, but on ultramafic areas. So I went to a place that is a quarry that is here signaled by this red circle. It's a serpentine quarry that was used to obtain rocks, ultramafic rocks, to use them as ballast for the road construction while the mine was operating. When I visited this mine, I could see that this is a photograph of the mine, and all the red arrows point to one tree, one species of tree that is called Keutostoma terminale. So when I visited this quarry, you can see in the background that is poorly vegetated, but most of the trees growing around there and in the lower part of the quarry belongs to one species that is Keutostoma terminale, that belongs to the Casuarinaceae family. And then now let me to make a small parenthesis to talk to you about Casuarinaceae. That is also another difficult term I have in the name of my project. So Casuarinaceae is a family of plants, of trees, that belongs to the angiosperm group. They are not pines. They seem like pines, but they are angiosperms. They are flowering plants. This family, Casuarinaceae, is divided in 4 genera. You can see in the image in the bottom left, this is a tree, a phylogenetic tree of the 4 genera of Casuarinaceae. And we can see that the more ancient genus is Gymnostoma, then appeared the genus Keutostoma, and the more recent genera are Allocasuarina and Casuarina. In total, the 4 genera are in total about 80 species, and most of them belong to the genus Allocasuarina. This family, Casuarinaceae, is distributed on the tropical in tropical Asia, Australia, and also in Madagascar. And in Sabah, we have 4 species that grow naturally in Sabah, and 3 of them, 2 from Gymnostoma and 1 from Keutostoma, grow on ultramafic areas. Here I can show you a couple of photos. The photo on the left and the photo on the center are from UITM campus, and there are 2 species of Casuarinaceae. This one is Casuarina cunninghamiana, this one is Gymnostoma sumatranum, and the other one is from Mammoth Copper Mine, is Keutostoma terminale. And they are known in Malay as Sempilau, but also in Dusun language they are known as Toguhu, and in Iban language from Sarawak they are known as Bru. So these species have very special characteristics. One of them is that they have extremely reduced leaves. You can see here the the blue arrows on the right. They point to the leaves. The leaves are really reduced. They are tiny scales, less than 1 millimeter. So, as the leaves are very reduced, these plants make the photosynthesis with the branches. They have long branches, green, and they make the photosynthesis with these branches. Moreover, they have the stoma, which are small holes where the plants can breathe through these small holes. So the stoma can be exposed, such as the photo you can see in the bottom. You can see like a white line along the branch. This white line are the stoma that are exposed. This is Gymnostoma sumatranum. But in the casuarina and the Keutostoma, the stoma are not exposed. They are hiding inside small ridges or small valleys that we have along the branches. You can see in the image on the left the structure of one of these branches. No? So you can see that along the branch we have depressions, or the surface can be flat or can be a depression, and if the depression is here, the stoma will be hiding inside. The flowers in these plants and the fruits are also reduced. So the flowers look like small cones. These are female flowers in Casuarina echinacea, the image on the left. The image on the center are the male flowers, very reduced. They don't have petals. They just have the stamens. And what you can see here on the image on the right are the fruits of the 5 species that you can find in Sabah. So on the right we have here on the right top right we have Casuarina cunninghamiana, which is cultivated in Sabah. In the bottom right we have Casuarina echinacea, which is growing on sand beaches in Sabah. And then in the center and on the left we have the 3 species that grow on ultramafic areas. The one with the small fruits in the center is Keutostoma terminale. The one with the huge fruits is Gymnostoma sumatranum, and the other with the in the left bottom is Gymnostoma nobile. So these 3 species can grow on ultramafic areas in Sabah. And the interesting thing is that these species are able to grow on very poor soils, ultramafic soils, because they have several strategies that all of them to obtain nutrients. One of the strategies is the presence of clustered roots. This is the photo on the top. These clustered roots you have here a normal root, and then you can see that the roots, they produce a huge quantity of very fine roots, small roots that are very concentrated. These clustered roots are structures that are specifically adapted to mobilize the phosphorus from the soil and to absorb phosphorus. So the ultramafic soils are very poor in phosphorus, but the Casuarinaceae, they have roots, specialized roots that allow to gather and to collect the phosphorus even from these soils. Moreover, the plants, they are very efficient when they have the branches, when they have the green branches. Before the branches fall down, they are able to recover a lot of nutrients from the branches, and then the branches get brown and fall down. So they are saving the nutrients. They collect the nutrients and they keep the nutrients in the plant. And also they have an interesting thing is that the Casuarinaceae, they develop symbiosis, root symbiosis with a group of bacteria called Frankia that are able to fix nitrogen from the air. So the plant, in summary, the Casuarinaceae, they are able to collect nutrients either because they have adaptations, either because they have symbiosis with bacteria that can bring the nitrogen from the air into the plant. This symbiosis is also interesting because the different genera in family Casuarinaceae establish symbiosis with different groups of Frankia. So in Gymnostoma, as you can see in the figure on the left, the Gymnostoma established symbiosis with Frankia from the cluster 3, a group of Frankia. But the genera Casuarina and Allocasuarina, they establish symbiosis with a totally different cluster of Frankia. And interestingly, Keutostoma, that is a genus that is placed between Gymnostoma and Casuarina and Allocasuarina. We don't know until now, no one has studied the symbiosis in Keutostoma, so we don't know which kind of bacteria, which kind of Frankia are in symbiosis with those trees. So this is quite interesting. Well, so coming back to the query, after this parenthesis on Casuarinaceae, As I told you, in the quarry, in the serpentine quarry in Mammoth, the dominant species was Chaetostoma terminale. And what we could see is that each tree creates around them, the trees created small islands of litter. You can see, and I'm pointing with a cursor, you can see we have in the photograph, on the left, we have several Chaetostoma trees growing there. And you can see that around the trees, on the soil surface, there is an accumulation of litter. This litter is made by the branches of the plants that fall down and get a lot of time to get degraded. And when the plants are small, these litter layers can be a couple of centimeters thick. But below the canopy of all trees, you may have thick litter layers that can reach like 30 or 40 centimeters of litter in different steps of degradation. And the interesting thing is that we observed that several species, such as this Cyperaceae on the photograph on the right, they develop the roots, they colonize the islands of litter. I mean, we thought that maybe those trees are creating islands of fertility, islands of litter, that the litter creates conditions with organic matter and humidity that some plants can germinate there. So maybe these trees are acting as nurse species. I mean, the Sceutostoma or the Casuarinaceae, they are able to grow in these very poor soils, and they collect nutrients from the soil, and they connect nutrients with the bacteria from the air, and they produce the litter, and the litter creates conditions that allow the colonization by other species. So we did a study in the quarry. You have an image of the quarry, and also we compared this quarry with a natural soil inside Kinabalu Park, close to the Bulu River. In both places, Chaetostoma terminale was the dominant species, and in both places we can find that below Chaetostoma terminale canopies there was an island of litter. And in each one of these places, well, we published this research on 2018. The thing we did in this research was to collect in each one of these places, in Bulu River and in the quarry, in Mammoth Copper Mine, we collected soil without any tree. Then we collected soil close to a small tree, to small trees. Then we collected soil close to younger trees, and then we collected soil close to very old trees. And we observed the changes in the soil conditions and also the changes on the vegetation along this age gradient of the Casuarinaceae trees. What we observed was very interesting because, as I told you before, the non-vegetated soil was very poor in nutrients. Imagine that, for example, here we have either in the quarry or in the natural soil from Bulu, the non-vegetated soil, the concentration of essential nutrients such as phosphorus or potassium were really low, really low. So very stressful conditions for the plants. Moreover, the pH was quite extreme. Look at here that pH in the quarry was around 8, and the pH in Bulu River in Kinabalu Park was around 9. These extreme pH are also very stressful for plants. However, when we measure the conditions of the soils close to mature trees, we saw that the nutrient concentration increased very much. And for example, we passed from 0.03% of nitrogen to 0.12, that is almost 10 times more. Or in the case of Bulu River, from 0.01 to 0.36. In other nutrients such as potassium or phosphorus, there was also an increase of nutrient concentration. Finally, we also observe FDA is a parameter of the biological activity in the soil. We observe that in the non-vegetated soils, the biological activity by soil microorganisms was very low, but on the soils close to mature trees, the biological activity was much more bigger. it seems that the soils close to mature trees, the soil functions, the soil nutrients and the soil functions, the soil biological functioning is recovered. So we can see that the Chaetostoma terminale or the Casuarinaceae trees are able to recover the soil function, but we observe a little effect on vegetation. And this is why of the litter. The litter creates good conditions for germination because they have a lot of nutrients there and keep humidity, but only those plants species which have small seeds that are able to fall inside the litter are able to germinate, because these litters, you can see here a cut in the litter of Chaetostoma terminale. The litter is very fibrous, so it does not retain the water. When it rains, the water cool down, get down in filters inside the litter, so the surface of the litter is always dry, so the seeds cannot germinate on surface, and the humidity is below. So only the seeds that are small enough to fall and to pass between these small species between the litter and reach a place inside the litter layer are able to germinate. So the litter poses a problem for the use of these plants of Casuarinaceae plants for the restoration of ultramafic soils. We need to improve the degradability of the litter. So with these previous results I obtained on 2018, I prepared the Casuabiota project, which has as a long-term objective this question. We want to know if we could use these Casuarinaceae species as tools to restore degraded ultramafic areas or other metal-rich soils such as Mammoth Copper Mine. So I prepared this project, Casuabiota project, using as model species the 3 Casuarinaceae trees we have on ultramafic areas in Sabah: Chaetostoma terminale, Gymnostoma nobile, and Gymnostoma sumatranum. If we want to know if the Casuarinaceae can be useful for the restoration of the soils, we need to know 2 things. The first thing is that we need to know a bit more about the bacteria. We need to know which frankia live in the roots, or if these frankia living here in metal-rich soils have any special tolerance to this to the soil conditions here, or if they just are protected by the plant and they don't tolerate the extreme conditions in the soil. So to answer these questions, we are developing 3 different lines of research. On one side, we are trying to identify the frankia by DNA extraction, PCR, or certain genes and sequencing of these genes. Moreover, we are studying the microscopical structure of the nodules where the bacteria are living in the roots, to know more about the morphology of the frankia and the presence of nitrogen-fixing vesicles and other structures. And finally, we are trying to isolate the frankia in pure culture to develop experiments of tolerance to metals. Regarding the nodule structure, we have collected nodules in several places of the 3 species, in several places in Sabah, and we have processed the nodules. We clean the nodules, we have cut the nodules, and with hand, we cut the nodules in thin sections and semi-thin sections, and we stain the nodules using the cuttings with nactophenol blue, and try to observe the structure of the nodules using light microscopy. So you have here some images. On the top, you have images of the nodules of the 3 species: Gymnostoma nobile on the left, Gymnostoma sumatranum on the center, and Chaetostoma terminale on the right. You can see that each nodule mass can be quite big, can reach 10 centimeters of diameter, and each of these masses is composed by lobes. These lobes, I mean, each one of these nodules, they have a correlate structure. They are branched structure with the bacteria living inside. The photos at the bottom show which is the shape of each one of these lobes in the nodules. You can see that the morphology of the lobes is quite different between Gymnostoma novile, Gymnostoma sumatranum, and Cautostoma terminale. In Gymnostoma novile and Gymnostoma sumatranum, the lobes are thin, about 1 millimeter or 1.5 millimeters. And in Gymnostoma sumatranum, they are much branched than in Gymnostoma novile. However, the lobes in Cautostoma terminale are thicker. They have like 4-5 millimeters. I mean, they are one of these lobes are thicker than the lobes in Gymnostoma novile. So the morphology, the external morphology is different. When we do cuttings and we observe the distribution of the bacteria, you can see here several cuttings made by hand and stained. They are images, microscopical images. So on the left, we have 2 images from Gymnostoma novile. You can see the first photo on the top on the left is a longitudinal cut of one of the lobes, and you can see that we have on the apex of the lobe, we have the meristem, that are small cells that are developing and dividing and growing. This is the part where the nodule grows. And then you can see in this part of the nodule, you can see like blue dots. These blue dots are cells which carry inside the Frankia bacteria. And the other image we have here on the center, top center, is a transversal section of one lobe of Gymnostoma novile. And you can see in the center, the V indicate the vascular tissue, and all these blue dots around are cells infected by Frankia. The structure of Gymnostoma sumatranum is quite similar. Each lobe has in the middle a vascular tissue, a vascular bundle with all the vascular, the xylem tissue and the phloem. And then around them, you can see the Frankia. They are marked with an F. These are the Frankia infected cells that can be seen as blue dots or gray dots. However, in Cautostoma terminale, the morphology of the lobes are quite different. And instead of having only one vascular tissue, they have a lot of them. So they have a deformed structure which much more vascularization, and this is why the lobes are thicker than the lobes in Gymnostoma novile and Gymnostoma sumatranum. Moreover, the observation and higher amplification in the microscope, we can see that the infected Frankia cells are quite big, and the infected Frankia cells are marked with an F, and they are quite bigger than the cells around, and they are surrounded by thin cells that make a kind of capsule to contain the infected cells with the Frankia. In the image on the right, on the top, you can see a line of Frankia infected cells. These cells, they have Frankia inside, and the arrow is signaling some IFE. The Frankia live inside the plant cells, and they can grow across the cells, across the cell wall that divides the plant cells. What we observed also in the tail, in the cells infected by Frankia, the black arrows point to small spherical structures that we think that they are vesicles where the Frankia fix the nitrogen from the air. But we are now, not sure if these vesicles are really vesicles or other plant structures. So in this moment, we are preparing a different procedure for the obtention of better sections of Frankia, of nodules to study the morphology and the distribution of the bacteria. An interesting thing that we found is that when we observed the roots and we tried to find the nodules, we observed other small structures on the roots. You can see here in the 3 photos on the left, on the top of the left, you can see that in the roots we observed other small structures that are not Frankia nodules, but they are quite similar to mycorrhizal nodules from other Gymnostoma species from New Caledonia. So This is also interesting finding because it indicates that these trees are able also to establish symbiosis not only with Frankia bacteria but also with other fungi that are important for the obtention of nutrients from the soil. Regarding Frankia phylogeny, we collected the nodules from the field, we prepared the nodules, and we extracted from the nodules, we extracted DNA. And we made a PCR, we amplified a gene from the Frankia that can be used as the PGK gene that can be used for distinguish between Frankia strains. After we obtain the amplifications, you can see in the image on the right bottom is electrophoresis gel where you can see the PCR products. Once we obtained the PCR products, we sent to a sequencing company in West Malaysia, and they provided us with the sequence of this gene in all the extractions we made. And we compared our sequences with the sequences of other strains, and we obtained very interesting results. What I am presenting here, you can see here, you have a tree with several branches. This is a tree, and all the small letters at the top represent different Frankia strains or isolations. As I told you, Frankia, the Frankia bacteria is organized in several clusters. The cluster 1 that is on the right, the cluster 3 that is on the left, and in the middle we have two clusters, the cluster 2 and the cluster 4. These are very different groups of Frankia bacteria with very different sequences. And on the image you have on the left bottom, this is a reminder that in gymnostoma, or the literature, the bibliography said that in gymnostoma there were only cluster 3 Frankias, and Casuarina and Alacasuarina, they have only cluster 1 Frankia. The interesting thing is that we observed the blue circles mark the position where we have our samples from Sabah. We observed that the 3 species, Gymnostoma nobile, Gymnostoma sumatranum, and Chaetostoma terminale, all of them have Frankia bacteria that belong to cluster 3. Another interesting thing is that these strains we obtained, they are not all together in one place of the tree. They are separated in different groups along the branches of the tree. This indicates that they belong to potential new species. So probably we are dealing with between 4 or 5 different new Frankia species, which is quite exciting. But the most interesting thing is that we have found that Chaetostoma terminale, but also Gymnostoma sumatranum, they have also Frankia from the cluster 1. And this is also interesting because it's totally unexpected. It is totally new. We didn't know, for the first time, we know that Chaetostoma terminale has symbiosis with Frankia from cluster 3 and cluster 1, but Gymnostoma sumatranum is supposed to have only symbiosis with cluster 3, but now we know that Gymnostoma is also able to establish symbiosis with cluster 1. This is very, very interesting and totally unexpected. Continuing with our research with Frankia, we are also trying to isolate Frankia bacteria from nodules. And for that, we obtain the nodules, we clean them, we cut them, we sterilize them, and then we transfer, after crushing, we transfer them to these sterile tubes with a specific growing medium. The problem with Frankia is these bacteria, they grow very, very slowly. So we need to wait for several weeks or even months to see if they are able to grow. At this moment, the image you have on the right, you can see several tubes with nodules at the bottom, and we have now waiting. I mean, this is a bacterial culture. This is an experiment of isolation that is currently running in UITM Sabah, and we hope that from the more than 100 tubes that we prepared, we can obtain at least we can obtain one or two different strains. But this is still a work in progress. And I told you that if we want to study to use the Casuarinaceae, we need to study the bacteria, but we also need to study the litter and how to degrade this litter faster. So I have several questions. First one is, how is the composition of this litter? Which are the organisms, especially the fungi, involved in the decomposition of the litter? And if we can use other organisms from the soil, especially earthworms, to accelerate the decomposition. To do that, I am developing like different lines of research. On one side, I developed a vermicomposting trial that I will talk about this right now. We are also trying to identify the fungi in the litter following a fungal metabarcoding strategy. And also, we are studying the earthworms living on the litter in the field. For the vermicomposting trial, we did a trial where we put small bags with Casuarina accidifolium litter yeah. Well, so we did an experiment of the litter decomposition with some earthworms. So we put litter in a small. We put the litter in small bags, and these bags inside in boxes full of vermicompost. And in some of the boxes, we put some earthworms of the species Seseni andrei, that is a species widely used in vermicomposting. And we have followed the decomposition. The thing we observed is that after almost a year of decomposition, we still have around 50% of the litter. So the decomposition is quite slow. And we observed also that the number of earthworms and the biomass of earthworms with time decreased. This shows that the earthworms, they don't like a lot the Casuarinaceae litter, and that we have to try other strategies. And this is an image, but I will pass. So as we observed that the earthworms didn't have a lot of effect on the litter, we try a different strategy. So we built a bigger experiment in Sabah, in the UITM, where we put big boxes with soil from mammoth, and on top of the soil, we put some litter. And some of this litter, we treated the litter with a solution of vermicompost. So we are trying to observe if the application of vermicompost is able to accelerate the decomposition using more natural conditions. This experiment is currently running, and we will harvest it next March. Finally, the experiments or the study in fungal and earthworms are currently are work in progress. Regarding the fungal studies, we tried to extract DNA, and we did a first sequencing test, but we obtained we had contaminations of DNA during the extraction of the DNA from fungi. So we are trying to develop new methods, and for the earthworms, we hope to obtain more results soon. So to conclude, the most interesting things we have found until now is that the Casuarinaceae from Sabah, they have provided totally unexpected symbiosis with Frankia from cluster 1 and cluster 3. So it seems that after our results, we have to rethink about the evolution of the symbiosis between Casuarinaceae and Frankia. So this is quite exciting and interesting. And regarding the litter, it seems that vermicomposting with earthworms is not very efficient. So we will see after 1 year if the application of vermicompost can help us to degrade the litter. So thank you very much. And I'm open now for your questions. So if you have any question. Well, I'm sorry because I'm not sure if I was able to transmit all the things. I talk about very different things, and well, just let me know if you have any question. I will be happy to answer. No questions?
One of the questions was, how do you recognize ultramatic soils from the students?
From the what? From the?
From the students. From our
From the students,
today, yes.
Well, to identify ultramafic soils, I mean, we rely on the previous information we have. I mean, we know where we have ultramafic soils in Sabah because there are geological, I mean, the geological survey or geological service from Sabah and from Malaysia. They have quite good maps. But when you have to also, when you go to ultramafic soils, you can distinguish them also because of some properties. The ultramafic soils derived from peridotites in Sabah, they turn into laterites. So they are very red soils, which are mostly composed by iron oxide. So you have like soils that are very thick and totally red, very red, with only a thin layer, black layer with nutrients on the surface. And the rest of the soil is very deep soil that is red, intense red color. And the other ultramafic soils you have in Sabah, derived from serpentinites, these soils, they have a special color. They are like gray or greenish gray. If you go, I don't know if you have been ever to Mammoth Copper Mine. For example, in Mammoth Copper Mine, it's a place where they have two kind of soils. They have one soils derived from porphyry, is the soils they exploited to obtain the copper. But then you have also, in contact with the Mammoth Copper Mine, you have ultramafic soils, and the color of the soil is amazingly green. It's green or gray because of all the rocks that gave birth and the minerals that gave birth to the soil. So sometimes just for the observation, you have the soil in your hand and you can distinguish them with the color. And especially there are some species, especially the hyperaccumulators, that in Sabah they only grow on ultramafic soil. So if you find a Phyllanthus valgoi, for example, or other species that are hyperaccumulators, you are sure that you are on ultramafic soils. I have seen one question here about the application we use to construct the tree. You have several options. One is, I want to write here, one is called Mega. Mega Mega is a program, is a software where you can use to align sequences and to construct trees, and it's free of use. And also another software I also use is called CView. I'm writing on the chat. I wrote the names of the two software, and both of them, CView is developed by some researchers for the University of Lyon in France, and it can do several things, but Mega is more complete. And both of these programs are free to use. I mean, you can download. You just look on the internet, on Google, and you can download them for free and use them without any restriction.
Thank you.
You are welcome, Elden.
Hi, how are you?
Hi. Well, I'm fine. Happy to share with our friends.
Yeah. So I have question. Another last question about Mega. Do you use the Mega 6? What version of Mega?
Yeah, I use Mega X because I'm working with the operative system I have in my computer is Linux, is not Windows. So I use Mega X, that is a version especially for Linux system.
Oh, Linux system. Okay, thank you. So I try to use that. Thank you.
Yeah, You're welcome. But if not, CPU is quite easy to use, and it has also good options. I mean,
Oh, okay. Thank you.
you're welcome. Any other question?
Hello? Okay. All right,
Yes.
Dr. Tino, thank you for that response. We have one last question for our webinar today. One of our participant questioned that they said, I see that you are always in the wild while doing your research. How do you deal with bugs and mosquitoes?
Oh, well. Well, actually, I think the problem, well, I mean, I'm concerned about mosquitoes, but mostly the problems with mosquitoes is more in the coast. When you are working inland in the forest, I rarely see the mosquitoes. That is, I mean, the mosquitoes is the thing that I'm concerned more. Then we have the problem with the sorry, with the oh, with the eye, how to say, with the annelids, no, these worms that suck your blood. The tongue one, I know it's tongue one, is, well, these other bugs that suck your blood, I cannot remember the name in English. Oh, the slice, no. Slice? No. Well, but they are not dangerous. I mean, you just remove them and a bit of repellent and it's okay. No problem.
Do you mean the leeches? Or people
The leeches, yeah, the leeches, sorry. Yeah.
People would call it inta, here.
The leeches, yeah. And with the leeches, but there are some places that are especially rich when you go, for example, to Bulu River inside Kinabalu Park. The way to there are lots. And at the end, you finish with a lot of them on your body, but, well, you just remove them and, well, they are, can be a bit annoying, but not dangerous, right? You cannot get any disease from them, so it's okay.
Oh, okay. Thank you for that answer, Dr. Tino. So apparently mosquitoes aren't a big deal in the wild. So it's more about the leeches and all other.
Yeah, but mosquitoes, as I told you, I mean, especially if you work in the coast, maybe you can be concerned about mosquitoes, but in the forest, I rarely met mosquitoes. And sometimes when just a bit of repellent, then it's okay, not problem, no problem.
All right, thank you for that invaluable insights. So thank you all for your engaging questions, and a special thanks to Dr. Tino for providing such comprehensive responses. Before we go to the next session, we would like to direct the participants' attention to the chat because the link and the password for today's program is just posted. Please fill in your attendance in the link. Also, note that the Google Form link would only be open for 40 minutes. Only students with the correct password would receive college coupons. Right, Dr. Tina and all of our participants, as we near end of our webinar, we would like to invite everyone to participate in our photography session. We encourage you all to open your camera and smile for the picture. I would also like to invite my teammates to take pictures, and we will also like to encourage all of our participants and our students to open your camera for documentations.
Baju mah, Sophie, mul.
Participants, you can open your cameras.
They're sleeping.
I wonder. Maybe their cameras are working. Thank you for all the participants who opened their cameras. Right, we will take a picture. 1, 2, 3, smile. One more. 1, 2, 3, smile. Okay, thank you so much, everyone, for participating. Right. So thank you all for participating. We have now reached the end of today's webinar, Get to Know Nature. We sincerely thank you, Dr. Tino, for his remarkable insights and everyone who participated and contributed to the discussion. Remember to stay connected and continue your exploration of nature. Before we end, Dr. Tina, do you have any final remarks or final remarks you want to give?
Well, just I thank you to the students to organize this, and thank you to the people in the COMSAT, in Java, because without them this research would not be possible. Especially a special thank you to Dr. Y, that has been leading the research in Java, and well, I'm very grateful to her and also to other colleagues, Ms. Farnida, Ms. Yamina, Dr. Yolena, and others that have been collaborating with me. And well, thank you very much. Just keep your curiosity and keep exploring nature.
Right. Thank you so much, Dr. Tino. So have a wonderful day ahead, and thank you for joining us today. Before that, we would like to remind our participant again to fill in the attendance form, and please note that the Google form link would only be open for 40 minutes. Again, we would like to mention that only students with the correct passwords would be eligible to receive college coupons.
Well, bye-bye.
Right, Dr. Tino, bye-bye. Thank you so much for today.
Bye-bye.
Bye, Dr. Tino.
Hola, ¿cómo estás?
Bye-bye.
Thank you everyone for joining today. Please make sure to fill your attendance, and please make sure to get a correct password, because otherwise you wouldn't get college coupons.